Next, we have up Dr. Eric Gershwin, a distinguished professor of medicine at UC Davis. One of the things, we know with Dr. Gershwin, he's done a lot of research, a lot of study. We've supported him quite a bit in this as well. He's really devoted about finding a cure for PBC. But one of the things that's interesting too, as we'll touch on, is some of the interesting mouse models. But one of the aspects that I thought was interesting is why was the mouse so good at studying pathways, disease pathways? Because it knew how to squeak through the tough parts. And then one other thing, what do you call a mouse that can explain how PBC works? A spokes mouse for the scientific. Okay, that's enough. Okay, so next we have Dr. Gershwin coming up here and telling us a little bit more about unmet needs in PBC, how we can solve them, and some discussions here. So again, feel free to enter some of the questions into the app. Touch on the session that says unmet needs, and if there's some questions, we'll address those at the end and throughout as needed. So thank you, Dr. Gershwin.
Boy, Tom, that's a hard act to follow.
Yeah,
I got to tell you. And Dr. Vealing, who I've known now probably for at least 50 years, I've got to guess, maybe longer, 1967 or so, said I have to kind of introduce this by getting everybody kind of a bit enlightened, loosen things up. And I'm trying to think how can that be done? And I always give a different lecture each time. I'm not one of these people that gives canned lectures all the time. So I got to tell the story, I guess, of the lecturer and the chauffeur. If you've heard it or not, Dr. Vealing, John? So, there's a doctor went all over the country lecturing, giving the same lecture year in and year out, every state, every medical school. But as he got older, he couldn't drive. So he had a chauffeur, and the chauffeur would take him from place to place. So one day, and I think it was in upstate New York, maybe Rochester or Syracuse would be better, I guess, since I grew up in Syracuse once upon a time. And he said to the chauffeur, I'm too tired. You heard his lecture 100 times. I want the lecture given by you. So the chauffeur said, I can do it. I've heard your stories, your jokes. He got up there, gave the same talk this famous doctor had been given for the last 10 years. And he started to take questions. And he heard all the questions already. Everybody asked the same questions. And then some in the audience got up and they asked a really simple question, but apparently it had never been asked before. And there's the chauffeur standing up there, the doc sitting in the back, and the doc is wearing the chauffeur's uniform, of course, and the chauffeur standing up there in the doctor's uniform. And this guy asked the so-called doctor the question, didn't know the answer, and he said to the guy, that question is so stupid, I'm going to ask my chauffeur sitting in the back to answer it. It's a complicated joke, I'm sorry. Anyhow, I wanted to introduce really a lot of the things that we've been doing and have been doing. I cloned the mitochondrial antigen that's reactive with the anti-mitochondrial antibody in 1986. And I thought everything thereafter was going to be easy. And boy, was I wrong. But I think things are getting easier, and I think we're learning a lot more. I think we know more about the biology of PBC than we know about almost every other autoimmune disease. I mean, it's amazing to think about it. I really do believe that. And people that I've worked with and panels I've been on, they say the same thing. But yet, we don't yet have a cure, which means that we're missing something. Maybe we're just too stupid to figure it out. So let me talk about some of the things we're doing. Tom, do I move the slides, or do you move them?
Hit the arrow, the blue arrow, the green arrow.
This thing? No.
No, on the square box.
There we go. Okay. So first, let me tell you about UC Davis. UC Davis is actually now the largest of all the University of California campuses. We have more students and more faculty than UCLA, UC San Francisco. We serve an area which goes up to Oregon, all the way halfway down to Bakersfield, and all the way out to the western portion of Nevada. Patients even come to us from the Bay Area because they don't want to deal with the traffic of San Francisco. We used to be entirely on the campus, which has the best and the largest vet school in America. And now virtually everything is located in Sacramento at our University Hospital, which is the largest healthcare provider in inland California now, except for Kaiser. Here's where we are. A lot of people have never heard of Davis, but it's only about 18 miles from the capital, which is Sacramento, of course. The map of the campus, you probably don't care about that. This is our building. It's the Genome Center, and we take up half of the 6th floor of this building. We've had meetings. I know some of you have come to our meetings with Dr. Chris Bolus, who's our resident hepatologist, where we collect as much blood, feces, and saliva as we're allowed to do. Some of you may go back to the days when we did this at the Holiday Inn in San Francisco, and I've almost got sued by the company, who was very upset that I was doing that, where I had collected in their hotel. This is our group. As you can see, we have people from all over the world, predominantly Asian these days, because it is so hard to get Americans to do basic research, and people don't want to study difficult problems. So it's a lot easier to get someone to study hepatitis C than it is to get someone to devote their career to a disease like PBC. We've collaborated because I think to find a cure for PBC is going to take a multidisciplinary approach involving immunologists, molecular biologists, biochemists, hepatologists, amongst others. And I'll tell you that since I started this work, and it's now been more than 40 years ago, our lab has given DNA, sera, or mice for free to anyone who's ever asked, without any MTAs or any of the ties that usually people require. An MTA is material transfer agreement. So these days, you can't even go to the bathroom without getting permission from somebody if you want to do collaborative research. We always find ways around it. At least we try. I've trained over 100 people who are all over the world now. I'll give you some examples of people that have worked in our lab that now study PBC or other liver-related immune problems, large groups in Japan, Taiwan, in Korea, major collaborations now, both in Shanghai and Guangzhou, where I actually have meetings on a regular basis and actually help supervise a lab there. In Canada, Italy, extensively. Only one person in England. English are not famous for collaborations. Israel, France, many people in Western Europe, in Greece, Dimitri Bogdanos, who just become the new editor of the Journal of Autoimmunity, in Holland, in Spain, South America, and of course extensive people throughout the United States. So that's us. And what's happened in 40 years? So what have we learned, and how do we solve those unmet needs? Well, we've worked on both etiology to try and understand what the genetic basis of PBC is and what the environmental factors are. We know, and many of you participated in our studies, that you don't get PBC from genetics alone. It requires one or more environmental hits. And it may well be that there are multiple hits required, and it may well be there are many different multiple hits involved. And I'll talk about what we think some of those are. We've put enormous effort into animal models. And we've developed now, I'm guessing, 5 or 6 different animal models. And we keep trying to refine them because we really think that if we are going to find a cure, we need an animal model that's going to really recapitulate human disease. And that's not easy to do because in so many other autoimmune models, the mouse has not faithfully recapitulated what happens to our patients. And of course, we've worked on both prognosis and tried to introduce new treatments based on our animal models. So let's talk about etiology. So once upon a time, in a disease which we thought was simple, like rheumatoid arthritis, we thought, oh, maybe there's going to be half a dozen genes, and you'd know exactly what caused rheumatoid arthritis. And so the Human Genome Project got started. It costs $1 billion to sequence one person. I won't put it in the mic as to who that one person is, but I know who it is, and we talk about it off the mic. I don't know if Dr. Viewling knows who that so-called person was. But in any case, you can now sequence somebody for about $1,800, $1 billion to $1,800. Either way, there have been large studies throughout the world. Many of you have participated, where we take the genes from you and we compare that to the genes of someone who doesn't have PBC. Or we look at patients who have a sister or a twin, and many of you helped us with twin studies to see whether or not there's any genetics involved. And I have to tell you that although I am going to show you some clues, it's been a bust. And it's been a bust in PBC, it's been a bust in lupus, it's been a bust in rheumatoid arthritis, and diabetes, and MS. We all know there is a genetic susceptibility. The problem is it's not a simple one. It's very, very complex. And in fact, it may be more than just the DNA. It may be what's called epigenetic alterations, where your DNA undergoes methylation, as an example, and that will change how your ultimate immune response is. Most of our genetic studies now involve groups of people of 1,000 or more, in which, at least in one case, we have found a variant that's now been reproduced, in which we can say it's associated with a considerable higher risk. But it's not a home run, meaning I can't take blood from you and tell you, because you have this, you're going to get PBC. I can't go to a family with a mom who's got PBC and test her kids and say this is the clue as to whether or not her children are at risk. Unfortunately, we're not there. It is clear that at least with this one variant, we can try and relate it to liver function. One of the paradoxes to me in PBC is that we all consider this an immune disease. Our immune system is attacking ourselves. I tell people the immune system is like your car. You have a gas pedal and you have a brake. And that car can get out of whack in only a couple ways. You step too much on the brake and the car slows down and you get severe infections, because by slowing, your immune system isn't working, and that would be examples of people with immune deficiency. Or you got a car in which you're stepping on the gas too much and the car goes out of whack, it goes too quick, it attacks itself and it gets hurt. And that's an example of an autoimmune disease like PBC. And we can define the antibodies and everything. So why am I mentioning that? Because if all that is the case, PBC should not come back after a liver transplant. And I've argued this case with some very good immunologists, and many people are trying to come up with a solution to it. But the reality is it doesn't fit the normal scheme of immunology. Somebody won the Nobel Prize for something called restriction, MHC restriction. And without explaining what that was, if that guy really, or those two people deserved the Nobel Prize, PBC shouldn't come back after liver transplant, but it does. And that's an outlier that we continue to study. It's part of an unmet need, because I think by studying outliers, we'll be able to figure out a little easier what the big picture is about. Just as a summary of what the genetic studies have been, because I mentioned it because I know some of you have been involved. We can show this gene is a risk factor. We can show it is involved in a single nucleotide. And we also can look at function and show that it promotes cholestatic injury. But the translation of that at the bedside to help patients is just not there. One thing which has been true of every epidemiologic study done, both the one that you guys did, the first big US PBC study, which is probably at least 20, 25 years ago now, we found that women with PBC, before they got PBC, had a history of more urinary infections. And that's been true of similar studies in the UK and studies in Western Europe. Well, when you make an anti-mitochondrial antibody response, that antibody reacts with an enzyme called PDC, PDC-E2. And that's an enzyme involved, if you guys ever can remember your biology, involved in something called oxidative phosphorylation. And that enzyme is so important in evolution that people have it, dogs have it, cats have it. It's found in worms, it's found in plants, and it's found in bacteria. And so one theory has been that you have a urinary infection, you react with the PDC in the bacteria that's producing the urinary infection, but in some people it turns on you and reacts with your own PDC, and that starts the cycle of what the clinical entity of PBC is. And it's hard for me to point at all the various cartoons in here. I'll tell you that you can't be an immunologist without showing cartoons that nobody understands. But that's kind of the purpose of being an immunologist. But in any case, we started to work with a guy named Frank Jordan. Frank Jordan is he's now retired. He's a chemist back at Rutgers. And I like to find people who have never heard the question, because maybe they know the answer. But because they've never heard the question, they can't give you the right answer. And I went to Frank Jordan because he's been studying the structure of PDC in bacteria all of his career. And I went with him with a simple question. It sounded simple. And that is, can he come up with different components of PDC in bacteria? And can we ask the question of whether antibodies to the E. coli PDC come before antibodies to the human PDC? And again, this is kind of a summary of a big paper, but Frank developed a sequence for us, which he calls E2-3-LIP. It's called LIP because it's close to where the lipoic acid site is for the docs in the room. And yeah, we were able to find out, he was able to show that the antibodies are found against this E2-3-LIP early on in patients with disease, with PBC. Is that proof that E. coli is involved in producing PBC? Unfortunately, it's not. It's just another clue. And one of the reasons why it's so difficult is because I think PBC starts, I know PBC starts for years and years before anybody ever gets diagnosed. We did a study back in Italy where we collected, I'll have to make the number up because I don't remember, we collected several thousand sera from women who gave blood to the equivalent of the Red Cross in Milan. And we screened them all for AMAs, and we found a handful that were positive in women that thought they were totally healthy. And we followed them, and all 4 of them have developed over the next, 8 or 10 years, developed PBC. So the antibody comes long before, which makes it very difficult to find etiology. Suppose PBC is caused by eating a bad apple that you had in 2004. How would we ever know in 2024 what started it 20 years ago, particularly in a disease as uncommon as PBC? So those are some of the challenges we have faced in terms of etiology. I mean, clearly, as far as prognosis, UDCA has made an enormous difference. But one of the things that we've been interested in is what's the role of nutrition and high cholesterols and lipid metabolism in PBC. And Dr. Veely and I were talking about Ian Mackay a few minutes ago. And Ian Mackay has a son named Charles, who's a very well-known researcher in Australia now. And he's interested in these HAMSA diets, HAMSA diets. These high-starch diets that produce a lot of butyric acid in the gut. And he's published papers in Nature that they help people with diabetes. And so we started to use those diets to see if we can improve lipid metabolism, increase butyric acid. We used them in our animal models, expecting that the mice would get better. And in fact, we found out was that the mice got worse. And that's got us, even though it didn't get the result we wanted, it got us excited to learn a little more, and I'll show you where I'm going to come back to, about the role of diet and nutrition in making PBC worse, or maybe even in induction phase. We've also worked with the Chinese on treatments of interface hepatitis. And, Dr. Veely is know so much more about that than I do that I am embarrassed to even to bring it up. But our point was that at our suggestion as a rheumatologist, that's what I am, and I use immunosuppressives a lot more than the hepatologists do, that the combination of, oh, sorry, how embarrassing is that? That at least in interface hepatitis, the addition of immunosuppressives was helpful. And I bring it up only because I'm trying to show that people that I've trained that have gone throughout the world continue to carry the banner of how we can help people with liver disease. But I want to talk about the animal studies because as an example, you can't even begin to guess how much it costs to do animal studies. Would you believe animal mouse bills a month of $15,000 or more to take care of mice? But in any case, we developed a variety of models over the years, and I'm not going to bore you or go into them, but I'd like to tell anecdotes. And one anecdote's about a immunologist named Howard Young. Howard is director, he just retired, but Howard was director of the cytokine laboratory at NIH. And he spent his whole career studying cytokines. And Howard, I believe, is in his late 60s, maybe early 70s. And he is a real card-carrying immunologist, one of these people that everybody looks up to, editor of journals everywhere. And Howard was interested in the role of cytokines. Cytokines, these molecules involved in inflammation, I should tell you. In fact, when you turn on the TV, you can't watch anything without hearing about a new treatment for psoriatic arthritis or rheumatoid arthritis or colitis. Those are all molecules that block cytokines. That's how important they are. But Howard was interested in cytokines and how they might affect lupus. And he developed a mouse, which is called Airedale. You don't need to worry about what Airedale means, but Howard believed that by changing the metabolism of a common cytokine called gamma interferon, the mice would get overwhelming lupus. And he believed that understanding lupus really depended on knowing more cytokine biology. So he developed these mice that involved gene knockouts, meaning you knock out a gene selectively and then you breed. It's a very time and very expert-induced sort of procedure. He developed these mice and he started to study them and he gave up. The mice didn't get lupus. It was a waste of time. And then somebody, because it's government, NIH, you can't just sacrifice mice. Animal safety is, of course, very paramount important to all of us, and so animals will sit on shelves. And they started to get sick. They started to die. And so they did autopsies on them, and they had some form of liver disease. And Howard is a PhD. He doesn't know much about the liver. And so for whatever reason, instead of calling the liver docs at NIH, he called me. And he sent me to Sarah. And lo and behold, these mice had incredibly high titer anti-mitochondrial antibodies. And in fact, the mice that had them were the females. And they itched. And we looked at the bile salts of these mice. The mice had abnormal bile salts. And we think biliary bile biology is very important in understanding PBC. So Howard sent us the mice, and we've been studying those mice ever since. We were also very much interested in this issue of whether what's called innate immunity also affects PBC. I've always believed that the treatment of PBC, if it's to be effective, has to be given early if you want to block the disease from going on, and that after that it's going to require precision medicine based on what your own immune system is at that time. And I think that what we call the innate immune system is going to be very important to that, which is, by the way, why I think PBC comes back after liver transplant. And I'm going to talk about these other mice as we get there. But, I still argue that there are limited treatments. I want a cure that helps everybody. I don't want a cure that just lowers liver enzymes. We'll take everything. And I'm as excited about the PPAR agents coming out now as everybody else. But the fact is, they're not like the biologics we use in rheumatoid arthritis. They're not as precise. They're not going to give you the result that I want to see in patients with PBC. Here are the mice for those that are interested. Here are the Airedales on the left, the wild types on the right, and you can see the incredible degree of biliary inflammation. And when liver pathologists look at these mice, as far as they're concerned, they're almost the same as humans with PBC. And interestingly enough, when we do these genome-wide studies, the signature of the mice looks very similar to the signature of the humans. We started to use these mice. Well, we try to look for treatments. We try to treat them with JAK inhibitors, which are these oral agents that we use in patients with PBC. The readouts were anti-mitochondrial antibodies and cytokines and T-cell infiltration. And the end result is, yeah, these agents can significantly help, but they help best when you give them very early in disease. And of course, that may not always be the case. And the other issue about it is how early is early? Meaning that a mouse doesn't come to you, 3 months after they're sick. We treat the mice both before they get sick and after they get sick. And boy, we get a lot better results when you treat them before they get sick. So, that isn't the kind of result we want. But I think that JAK inhibitors do have potential to treat PBC. They're not yet used. It's very hard to get industry to be interested in using the drugs that they're making hundreds of millions, if not billions on, to treat a disease like PBC. Some of you remember my story when, and I won't say who the company was, but I flew to Chicago to meet with people at their request to see if I would get their drug at their cost. I wasn't even asking them for money, just get their drug at their cost to use experimentally. And I flew all the way to Chicago, and at the last minute they changed their mind, and I flew home, never had a meeting. They didn't care because the market for PBC was way too small for them. And if I found the toxicity in my experiments, it would totally wipe out or hurt their market. That wasn't true of all the companies. Without mentioning names, we studied both a variety of biologics, including drugs that target B cells, rituximab is an example. And again, we got partial responses, but nothing terribly impressive. And again, it seemed very strange, and in mice, they seem to work if you give it to people before you get sick, in mice that is, before you get sick, but not after. And that's been the case of virtually every biologic that's been tried in PBC, which is why I keep coming back to the fact that maybe the paradigm that everyone has been following is at least partially wrong. We divide our immune systems into adaptive and innate responses. Adaptive is what happens after a vaccination. You promote the immune system to make an antibody or a cytotoxic T cell that fights a virus. Innate responses are what protects you when you go out and someone coughs on you. You want something to work immediately, hours later. They involve cells in your airway, they involve the cilia in your airway, involve a variety of cell populations. We think that over a period of time, it's the innate immune system, I'm going to skip that is probably the most important. And that's why in the treatment of these mice, it works best when you give it early. I'm talking too long. So I want to jump to the next model, which Carol helped us a lot with. PBC has helped us a lot with. We had this theory that if we could take a mouse who gets PBC and change its bile to that resembling human bile, that we would change the natural history of PBC in the mouse. Let me explain that again. We have a mouse with mouse bile. We have a mouse with human bile. It's not entirely human bile, but it's similar enough. And we did that basically by trying to introduce some new genes to change bile salt metabolism. And the end result is that these mice with bile salts that are human develop a disease which is much worse than any mouse with PBC that we've ever seen, including fibrosis and cirrhosis. This is probably the most important experiment that I think I've done in the last 8 or 10 years. I know it's complicated to understand, but what it's trying to say, to me it's trying to say, that early on you get an immune, what we call adaptive response, but over time the bile salts play a key role. That theory is not mine. That theory was developed by a guy named Ulrich Boyers and others in Western Europe, and they've been saying it for years. I give them credit for sure, and now I'm trying to figure out a way to exploit it to help patients with PBC. We've also found, by the way, that by giving a high-fat diet, we can do much the same way to these mice with PBC. But I think the most exciting work we've done, and this was just published in Nature Communications about, I don't know, a week or so ago, less than that, are using CAR T cells, chimeric antigen receptor CAR T cells. So for those who don't know what CAR T cells are, this was a science developed by really outstanding scientists in Pennsylvania to treat lymphoma, leukemia, other cancers, in which basically, I'll simplify it, we take, or they took, the cells from a patient, they would incubate it with cancer cells, again, simplifying this, so that your immune system would become sensitized to your cancer cells. They would expand them, and they would inject it back into patients. And this is now approved therapy for diseases like lymphoma, a whole variety of other cancers as well, and there are positive experimental trials now in human lupus. We decided to see if that would work in PBC. And we're particularly interested in a subpopulation of T cells that also express an antigen called PD-1, which is part of something called checkpoints. And again, I think it's more than I would want to talk about here. But essentially, and I want to tell you, and it's in the paper, we had to do this work in China. And the reason we had to do this work in China is that it probably cost us at least $200,000 to $250,000 just for the work that was published in that paper. And obviously we just couldn't do that here. But in any case, essentially what we did was we isolated T cells. We essentially exposed them to this antigen. We then expanded them, and I'm really making this very simple. We showed that it could target the liver and that it successfully, enormously successfully, reduced disease in at least this mouse model. And this has not been done in humans with PBC. It is being done in humans with lupus as we speak. But I think it is a very directed, almost like a guided missile approach to target the cell that we believe is responsible for maybe early PBC. But it may not work, unfortunately, in PBC as it goes further. So I guess the summary of 40 years of work, although mostly highlighted in the last 8 to 10 years, are that we've worked on both genetic and environmental factors. We're very interested now in the role of nutrition and bile salts in altering PBC, and of course trying to focus on how do we selectively get rid of those autoreactive cells that are basically destroying biliary epithelium. This is a picture of us. Admittedly, this is a picture before the pandemic, and we're not quite as many as we were then, but there's still about a dozen to 14 of us. And one of these days, I hope to come here and say, we or someone else found the cure. Thank you.
Thank you, Dr. Gershwin. And a reminder to everybody, any session questions, feel free to put them within the app. Also, we have Leslie going around and asking some questions. So Dr. Gershwin, it's great to hear all the different ideas and all that. As far as implications and some of the takeaways that patients, care partners that are here can take away from some of the elements like the nutrition, cholesterol, or other elements, anything you'd recommend for them to discuss with their doctor now before?
Sure. Well, certainly I defer to your doctors. So obviously the University of California would really beat me up if I gave medical advice up here, okay? They can't afford it, okay? But having said that, healthy diets are important. I think having high cholesterol and PBC is potentially very harmful. Having high lipids is potentially very harmful. I think it's harmful for their PBC. I would not take a dietary supplement containing lipoic acid. In fact, I wouldn't recommend taking dietary supplements at all. Most of them are imported. You don't know what's in them. And some of them may not have what you think is in them, what's said on the box anyway. That's sort of the lifestyle that I would talk about. I'm encouraged by the number of small companies that are interested in PBC. I mean, hardly a month goes by when I don't get a call from somebody that has some idea about some new technology, and I think a lot of them are really exciting. I think that a lot of advances that are happening now in cancer, not just CAR T cells, are going to be extrapolated to treat people with autoimmune disease. I think one of the major unmet needs are people with advanced disease. I think another unmet need is trying to figure out why PBC comes back after transplant in some people, but not in others. And, you know, I think the answer is research, research, and research. I'm always discouraged by the fact that there are not many labs around the world that really will study PBC. You know, a lot of people write clinical papers, but in terms of actual getting their hands dirty, I think more people need to study PBC, and I hope that'll happen.
Great. And where's Leslie? Leslie, any questions?
Yeah, with regards to Urso, Allergan recently stopped manufacturing Urso Forte. And is there a new company manufacturing Urso Forte as a brand? And are there other companies, or how many other companies are there?
Now, I'm going to be like the doctor and the chauffeur, except in this case, the chauffeur is a world's expert. And I'm going to ask Dr. Vueling to answer that question.
Do we have a microphone? You want to take the microphone up to Dr. Vueling, see if he may know? It's on.
Well, it's an excellent question, but unfortunately I do not know the answer because you want a contemporaneous answer as to what's going on now. I do think that I can find this out, and I will be happy to try to answer it for you. But having ready access through our healthcare system and around the world to ursodeoxycholic acid is essential for the treatment of PBC, as I hope I convinced you this morning. So this answer to the question is quite relevant. I just don't know it.
Yep. And we'll try to do some research as well to help others out, because I know also AbbVie, I think, acquired Urso 250 and Urso Forte. So, but I'm not sure about their manufacturing. But as that was 20 years ago for me as the product manager for Urso 250. So don't hold me accountable. Any other questions that you have, Leslie? Cool. Just keep your questions as mine too. Just to remind everybody that we will have the doctor's panel coming
what
up.
time is that?
That is at, anyone? What's that? 3:30? 3:30.
Sounds good.
Okay, so we'll have Dr. Gershwin come back up for that, but thank you for your presentation as always. Great to have your information. And yeah, give another round of applause for Dr. Gershwin.